So we are very pleased to have today with us in our Bar-Ilan University Vision Science Seminar, Professor John Greenwood from UCL Experimental Psychology. Professor Greenwood has an undergraduate degree in psychology from the Australian National University School of Psychology. He then continued to a PhD there under the supervision of Dr. Mark Edwards, working on the representation of multiple directions of motion within the human visual system. He then did a postdoc at the Institute of Ophthalmology at UCL, working with Professor Steve Dakin and Professor Peter Becks, and then continued to another postdoc at the Laboratoire Psychologie de la Perception at the Université de Paris Descartes, working with Professor Patrick Kavanagh. After that, he joined UCL, where he is now faculty and a professor of visual perception in the Department of Experimental Psychology, where he is the head of the Eccentric Vision Lab. His research aims to understand visual perception and the way we see the world. His lab examines vision in typical adults and children, as well as in clinical disorders such as amblyopia, which is the lazy eye, infantile nystagmus, which is uncontrolled eye movements, and visual-led dementia. Specific topics of his include peripheral vision, crowding, and the recognition of letters and faces. His lab uses techniques that focus mainly on psychophysics, as well as computational modeling, eye tracking, and neuroimaging. He publishes in top leading journals as Current Biology, PNAS, Nature Communications, and Brain. He has given dozens of invited talks across the world about his research, and his research has received funding from multiple sources, including UK Medical Research Council, the Academy of Medical Sciences, and the Moorfields Eye Charity. So without further ado, John, we are very pleased to have you with us today to share your research, and thanks so much for joining us.
Thank you, Sharon. Thank you for the lovely introduction. It's a pleasure to be invited here to be part of the seminars. I've been following these since the pandemic, right? They started. They were a really lovely way to come together in that time, and I've kept up with the talks since then. So it's really nice to be able to present today. Let me share my screen and we can get going. Okay. There we go. Okay. I've lost vision of all of you, but hopefully you can see my screen. Let me know if not. Yeah, as Sharon said, I'm going to talk about the work that we have been doing in my lab, particularly in the last few years, where we've been focused on visual crowding effects, and in particular comparing the way that they affect vision in typical development, in typical adulthood, as well as in atypical conditions associated with clinical disorders of vision. So I want to start with a problem, which is that the visual world is busy. If you were to come to UCL here in London on the first day of term, then you would encounter a scene like this, which is full of buzzing, chaotic noise, and students trying to find each other in the crowd. You have an onslaught of information when you encounter these scenes. It's a lot less like this on the last day of term, of course, but you still, in that circumstance, have to make decisions about a great deal of information. If you were, for instance, trying to cross the road down here, then you also have various time constraints on that. So you have to make rapid decisions sometimes about a great deal of information that we really need to recognize what's out there in the world. And if you were designing a system to perform this task from first principles, then you might start with something like what we have in front of us all here, which is something like a computer monitor where there's equivalent resolution all the way around the visual field. And you could represent each of those objects with equal fidelity in order to perform that task. But of course, we have to perform these tasks with limited neural resources. We have to use the machinery that's available to us, and using these kind of calculations, you can make the comparison that if we took the highest resolution that we have in the visual system, the resolution that we need in order to read and so on, and applied that across the visual field, if we had that level of representation everywhere, then our brain, which is about 1.3 kilos on average, would have to be about 13,000 kilos. And so that's something like a truck-sized brain that we'd have to carry around with us every day in order to represent all of the aspects of the scene that are coming at us at any one point in time. Obviously, then we're limited by the size of our skull and the mechanics of childbirth and all those sorts of things like that. So the visual system makes compromises, and the way that you see these compromises take form. Oh, sorry, I can see the. Let me. No, sorry. Here we go. Let me just hide the loading controls. There we go. Okay, so the compromise that we see is what's known as foveation, and you see this at many different levels within the visual system. You see, in terms of the structure of the eyes, we have two frontally facing eyes. Both of our eyes are pointing at the same source. And so we're getting this duplication of evidence that's coming in towards us. That's a common arrangement that you see primarily in predatory species, where you focus all of your resources at the space in front of you. Then, of course, if you look at the structure of the retina, we have what's known as an acute zone, which we refer to as the fovea. So this is a heat map of the photoreceptors here, where the highest density of photoreceptors are concentrated in the foveal pit, which defines the center of our gaze. And then you have this rapid drop-off in all directions around that. Going through to the brain, of course, then you have what's known as cortical magnification, where if you take the visual field and you color code it into the rings like this, and then project that onto the flattened surface of primary visual cortex, or V1, then you see that there's a huge overrepresentation of the central parts of the visual field in comparison to the outermost periphery, where you have far fewer neurons processing those parts of the visual field. And so this is the trade-off that our visual system makes in order to represent that onslaught of information, that we have this one particular region, this acute region of high fidelity vision, and then a periphery that is impoverished in terms of its inputs. And so what I want to talk about today are the functional consequences of that arrangement, both in terms of typical vision and thinking about why we have this kind of arrangement, but also in cases where that goes wrong, which we see in clinical disorders of vision. So an obvious place to start here is to think about acuity. It's no surprise, of course, that your peripheral acuity, your resolution, your ability to see fine detail, is worse than your foveal acuity. If you fixate on this red spot here, then you should be able to read at least some of those innermost letters. If I asked you to read around the clock on this innermost circle. But if you're fixating on that red spot and I ask you to read the outermost letters, so to start at the top, for instance, and move around the clock with those outermost letters, then you should find that much more difficult. Of course, we know that the acuity is an issue of scale, and so you can overcome this by scaling those elements. You can make them larger, and now if you're fixating on that spot there, it should be at least a little bit easier to read those outermost letters, if not fairly trivial. So we can overcome those acuity limitations. But there's a great deal of work that's been done in the last 50 years, of which many people here in the audience have already seen or well familiar. Evidence arguing that acuity is not the sole limitation on peripheral vision, that we have to consider other processes in order to understand what's happening in peripheral vision. And here's an example of that from my daily experience, which is a task that I'm very often given, an urgent demand that I find a very particular piece of Lego. And so if I lived in a featureless void like this, that task would be blessedly simple. So for instance, if you're fixating on this cross here, you might be staring neutrally into the void. Then you can make out in your peripheral vision aspects of this Lego figure, right? You could, if I asked you what color his hat was, for instance, you could judge that and so on. But if you keep your eyes fixed on that cross there, and I bring in the more typical context in which we have to do this kind of task, then you'll find that is considerably more difficult. If you even have a sense of where that Lego character was, then you'll at least agree that it's much harder to identify. But more likely, you lost all sense of where he is in that scene. So this is a demonstration that the context of our context matters greatly for peripheral vision in particular. And this is the effect that we refer to as crowding. In the lab, we tend to study this using simpler elements. So we have a single letter on the left-hand side, or these Gabor elements on the right-hand side, where you judge whether the tilt is left or right or vertical. But if I ask you now to fixate on that lower spot, the white spot, and to tell me the middle letter on the left-hand side, or to judge the tilt of the middle Gabor on the right-hand side, then you'll find that considerably more difficult. And so you can already see, so this is crowding. It's defined as the impaired recognition of objects in clutter. And you can already see that this is not something to do with acuity. These are objects that you can see. They're big enough to see in isolation, as you could see in this top row here. And you take the same size letter, but you put other letters near to it, and the interference becomes much more difficult. So this is something that happens above and beyond your acuity limitations. And it's also something that affects your ability to say to identify objects rather than your ability to detect them. So Dennis Pelly has shown that if you change the task to a detection task, if you say, is there a letter there, and you interleave trials where there is no letter, then your contrast thresholds for detecting that letter are as good in isolation as they are when they're flanked. So people can say that something is there, but they can't say what it is, in other words, which is quite different to the kinds of issues that you have with resolution, when things become highly blurred, for instance. One of the nice features of crowding is that it's very lawful in the way that it behaves around the visual field. So it's typically minimal in your foveal or central vision when you look directly at things. And in the periphery, the scale of crowding increases markedly as you move away from the fovea. So if you had a situation like this, where this is your fixation here, if you have a target at a given eccentricity, given distance from fixation, then you put flanker letters around that, you'll get these crowding effects. But what you can do is to move them away to a point at which they no longer interfere. And now you have this interference zone that defines where you see the effects of crowding. And there's always a localized region around a given target where you see these effects. What happens when you move further out into the periphery is that this same separation here will now cause interference, and you have to move the flankers even further apart in order to stop that crowding effect. And Baumer's law or rule of thumb, depending on who you talk to, is that the scale of these interference zones is approximately about half of the target eccentricity. So if this is 10 degrees eccentricity, then you'd have to move the flankers about 5 degrees away. Although there's been a lot of development on that specific figure, the figure is a little bit lower typically, and you get all sorts of interesting anisotropies and variations as well. What's interesting about crowding, from my perspective, is its ubiquity throughout the visual system as well. So I've already shown you examples on orientation judgments and letter judgments and the way that crowding affects those. You also see crowding effects if you ask people to judge the position of objects in the visual field, or the position of features within those objects, rather. So these little cross-like elements, we ask people to judge whether the bar was above or below the center. And you find impairments of crowding there. You get impairments to depth judgments. These are meant to be like red-blue anaglyph stimuli, where if you're judging a line in depth, you get interference effects from clutter as well. You get judgments, you get impairments to judgments of hue for patches. You get it for motion when they have moving elements like these Gabor elements. And you get it for complex objects as well. You get face recognition is impaired by crowding and natural scenes as well. I've shown you the examples already for those kinds of effects there too. So when we think about the visual system and the way that these different modalities are processed in different parts of the brain, a common feature to all of them is nonetheless that you get these crowding effects. And so it's both the ubiquity of these effects throughout the visual system and the large scale that you saw with those interference zones that leads to a statement like this, that crowding is the fundamental limit on visual perception in the periphery. I mentioned the kind of ubiquity of these effects is sort of surprising given what we know about the visual system and the sort of distributed processing of these kinds of features, which has been something that we've been looking at in the lab in recent years. And one of the interesting effects here is that if you combine these judgments of color and motion, so like with these little cowhide patterns down here, that when they have both movement and color, so observers have to say that the target here moved up and to the right and that it was blue. You can set up different circumstances where people can be correct about the direction and incorrect about the hue, and vice versa, depending on crowding, depending on the the magnitude of the crowding. And so that you can independently disrupt these features, in other words. And what that suggests to me is that the processing that's driving these effects is distributed throughout the visual system. That it might be more profitable to think about crowding as something akin to a canonical computation. That it's a process that's happening throughout the visual system in a lot of different places as a core feature of those areas. That kind of idea to me fits with the mixed evidence for the neural locus of crowding. When you look throughout the literature in terms of thinking about visual areas that process these stimuli and where you see crowding effects, there are arguments that you see correlates of crowding in terms of things like cortical surface area in primary visual cortex. There are correlations between the size of those interference zone and the size of population receptive fields in area V2. You also see these same kind of arguments about cortical area V4. We've also shown a while ago that in fact, you see neurocorrelates of crowding in terms of the perceptual effects, which I'll talk about shortly, across all of these regions. That you get a rise in the amount of modulation from crowding as you move up through the cortical hierarchy, but nonetheless that there are correlates of crowding across all of these areas. And so that to me fits with this idea of crowding being a more distributed process, more of a kind of canonical computation that's happening in all of these regions. So what is it doing? We have to think about what the mechanism is that's underlying this. And initially, a lot of the models of crowding focused on the errors that occur, that people are wrong in identifying a target letter or similar. And the argument then was that the reason that you're making errors in this case is because the target is lost in some way, either through inhibition or through inattention, because your attentional spotlight can't hone in on the target, for instance. But that doesn't fit with the nature of the errors once you start to look at the way that they affect people's judgments. And so something that we did a while ago was to look at a continuous judgment of the appearance of a target. And so if you had a kind of arrangement like this with these Landau Cs, although we use little cross elements there, what you find is that when people make judgments about the appearance of that central target element there in the array, that what they tend to indicate is something that's intermediate between its true horizontal to the right here orientation and the orientation of the flankers, that this is sort of intermediate. And so those errors tend to resemble something akin to the average of those elements in those circumstances. And that's not a response bias because you can also produce these effects in other ways. So in this case, we used these filtered noise targets surrounded by oriented Gabors. And what you find is that people behave as if the noise appears oriented to them. And you can produce all sorts of effects like tilt after effects and so on that are consistent with a real oriented signal being located in that region. So it's a change in appearance rather than a response strategy that's driving the errors in crowding. That the target is being made to look more like the flankers. Now, these kinds of errors are well explained by a particular class of model, which I'll call population pooling models. And really broadly, this is just the idea that when you represent a given feature dimension, like orientation or motion or so on, that you have along that dimension, you have the responses of an array of neurons. And then when you look across that population, you might have something like a Gaussian distribution, and we take the peak of that distribution as our percept. If you then surround that target by flankers, which have a slightly different appearance to the target, so the distribution is offset, then when you forcibly combine these two distributions with some weights to modulate how strong those effects are, then what you tend to get are things like this, where the peak of this distribution is now intermediate between the true target value and what you have in the surround. And so I'll talk about the way that we implement these kinds of models a bit more detail shortly. But the intuition here is that crowding is the unwanted combination of the target and flanking signals within a given neural population. And it's this idea that to me starts to speak to the idea that I raised in the beginning, that crowding is part of the adaptations that we have to deal with the sort of onslaught of information that we're faced with. And you see this built into things like the computational model by Freeman and Simoncelli, who took this idea of a pooling or averaging within receptive fields and applied it to a model across many different dimensions. So the model has effectively receptive fields that grow in size with eccentricity. And what they're computing within each of those within each of those receptive fields is the average color, the average orientation, the average phase, and so on. And so these are the kinds of images that you get out from the model. I'm sorry for the kind of warlike imagery here, but it gives a nice example of why things like camouflage work. And so you can see the model has a fovea that's denoted by the lines, and when the model is foveating on the soldier, the head is very clear. But when the model foveates over here, the soldier gets warped into the background because the summary statistics that characterize him are very similar to the house that he's standing adjacent to. And so the pooling across space gives you all of those properties, but it's not that the model sees nothing in its periphery. You can always make out things like the boundary between the sky and the houses. You can always find the door, for instance, because the summary statistics that characterize that door are very different from the surrounding regions. So the idea then is that this could be an adaptive way to represent these information-rich images simply with limited resources. That when we are faced with the impoverished inputs that we get from the periphery, then you summarize that into a gist, which gives you enough information to guide your eyes around, and you can locate objects of interest using that fine detail fovea. So all of that to set up the picture is as to the theoretical framework that we work within. But to me, that also then raises the question of why Crowley becomes elevated sometimes in foveal vision. And so this is something that you see across a range of clinical disorders. It's a strong feature in strabismic amblyopia, which is lazy eye that Sharon mentioned in the beginning, where one of the eyes is deviated either inwards or outwards. And in the amblyopic eye, you see elevations in foveal crowding. So even when they look directly at objects with their amblyopic eye, then they show these crowding effects. You also see crowding effects in cases of infantile nystagmus. So this is a disorder characterized by uncontrolled eye movements, and they behave as if they have these crowding effects in their fovea. You see it in cases of cerebral visual impairment, which is a kind of catch-all condition for damage to the brain, either during pregnancy or birth or in early life. And there you see elevated crowding effects in the fovea. You also see it at the other end of the lifespan in posterior cortical atrophy, which is a subtype of Alzheimer's disease characterized by visual symptoms in the first instance, and they show elevated crowding as one of their symptoms. On the other side of things, you also see elevations in peripheral vision at times. So there's arguments about whether this is the case in dyslexia. There's evidence in dyscalculia, which is the numeric form of dyslexia, and even in glaucoma, there's demonstrations. You heard from Mi-Young Kwon a little while ago about her work there as well. But take a moment here just to look at the diverse etiologies of these conditions. So you have what you would comfortably call things like ophthalmological conditions. You've got these kind of more neurological conditions, and yet all of them have this kind of common symptom at their core, this elevation in foveal crowding, which is quite a striking observation to me. The other thing that is worth paying attention to here is that the majority of these conditions are developmental disorders. These are disorders that have their onset during the early years of life. And so to get a clue into what's happening here, we should really be looking at the course of typical development as well, which is where I'll start. So it's been known for a while. It was great to see Jan Atkinson is here. She was instrumental in demonstrating that these foveal crowding effects are also present in the course of typical development, and that this is something that occurs later in life as well than the development of acuity. Acuity becomes adult-like around the age of 5 to 6, and there's a lot of evidence that crowding continues to be immature after that point, that it needs longer to develop. It suggests that there's a prolonged fine-tuning of our spatial vision over a long period of time. And that in turn may explain its prominence in developmental disorders, that if things are slow to develop, then they may be more prone to disruption at the point where these developmental disorders come online. So this is similar to Jan and Oll's ideas about the dorsal vulnerability hypothesis, for instance. The problem here, though, with crowding is that the estimates of these the point at which crowding becomes adult-like vary widely through the literature. So this is a figure from our recent paper where if you take estimates from papers throughout the literature, some of the authors are here. Then what I'm plotting here is the red bars are ages at which crowding is significantly elevated relative to adults, and the gray bars are where it becomes adult-like. And so you can see some estimates suggest that the transition is somewhere around 4 to 5. Some of them are looking at like 6 to 7. Some of them even later, and then others where there's aspects of crowding that are still immature, even at the age of 11 or 12. So there could be quite a protracted development of these abilities that's happening there. When you look through the various stimuli and procedures that are used, though, there's a variety of issues that you see. Some studies measure these things with stimuli that are not quite optimized for children, raising questions about whether the children are responding in a way that is reliable. Some of them use methods that we know that we've subsequently found are suboptimal to measure crowding, like about the spacing of the elements and so on. And so what we wanted to do was to measure these abilities, firstly using child-friendly measures, which we've been using for a number of years, but secondly using a number of procedures that are optimized to measure crowding. And so what we used was this VAC-MAN stimulus, which is this is visual acuity man, and VAC-MAN wants to eat the ghosts, so the children have to tell us which ghost he's facing using the orientation of his mouth there. And they can do that using the direction or the color or whatever they're comfortable with. There's a few different response options there to make it good for them. And then we can turn this into a crowding task by making the flankers now achromatic and bringing them closer. We use tight spacings because that's optimal to measure crowding. And we use a scaling approach, which I'll explain shortly, within this gamified context. One of the concerns we had is that children aren't necessarily sure which of the elements they're supposed to be responding about. And so the idea with adding characters into there is to hopefully make that clear to them. And we include catch trials and all those sorts of things as well. Now, the scaling procedure is that we scale the elements all up and down. So if you do that with an isolated element, that's what you do to measure acuity. So if they're correct, you make the object smaller. If they're incorrect, you make it larger. And then what you can do is to do the exact same thing with crowding. Now you have the flankers present around the target. If they got this particular trial incorrect, then we would make them larger. And if they get it correct, then they make it smaller again. And so the logic here then is that once you've measured your acuity threshold, you know that anything above that is not a limitation of acuity, but rather something to do with crowding. And so any elevation of that threshold beyond the acuity level is an indication of crowding. It's not that you necessarily need to make objects bigger, it's that you need to make them to shift those objects apart so that they move outside of that interference zone. But what it gives you is two independent measures. You're not dependent on your earlier estimates if those were noisy or problematic in other ways. This is the kind of data that we get. We measure percent correct recognition as a function of the gap size. And remember, everything is scaled, so we can use that to infer things like the size of the interference zone. This is example data from a 6-year-old child where you can see the unflanked performance is the dark blue points, and that very quickly moves up to ceiling. And then when you add the flankers, they're incorrect for a few more sizes, and then you get this more gradual increase up to there. We can take the threshold as 62.5%, and that gives us the gap size threshold that we can directly compare across those two conditions. And we're going to do that with typically developing children between the ages of 3 and 13. We managed to get 119 participants there. This is the kind of data that we get. So what I'm plotting here are those gap size thresholds, how small objects can be to still perform the task reliably. The unflanked condition gives you the acuity thresholds, and you can see that relative to the adults out here, that performance is elevated in this 3 to 4 year range. So the points here show you individuals, and these bars show you the age bins that we use to compare the groups. You can see this is elevated relative to adult levels, but then by the age of 5 to 6, acuity becomes adult-like. That replicates prior studies. What we find with crowding is that you get these strong elevations in the youngest children, which are still there to some extent in the 5 and 6 year olds, but then by 7 to 8, these levels are adult-like and they remain so thereafter. So by our hands, crowding is still becoming mature at an age later than what you see for acuity, but not as late as some of those prior estimates. Now, with the scaling approach, the beauty of this approach is that we can compare our estimates of crowding with those from prior studies. And so one of the issues here in making comparisons is that there are as many different metrics used to measure crowding as there are studies of crowding, more or less. Some people take the multiples of crowding relative to acuity. Some people take the spatial extent. Some people measure the edge-to-edge separation and so on. But we can use these estimates to convert our values because everything is scaled, and to convert prior estimates into the same space. And so this is what we did here, where we took 13 prior studies, including the current one, so as well as the current one, 14 studies in total, and just put them into the same range. So we use our data set as a normative value, and then align all of the data sets in that way, and compute this normalized estimate of crowding. And what you see is that relative to the adults down here, this is just flanked performance now, but you see very strong crowding in the 3 to 4 year olds. That's still present in the 5 to 6 year olds. But then from the age of 7 to 8, this is no longer significantly different from the adult levels, and that remains so. You get a bit of a dip in the mean here. That the mean is the black dots there, because there's so few studies that measure performance in this range. And so, although you see some variability in prior estimates of crowding, the center of mass certainly follows a very common trajectory. It suggests to us that there's an overall trend for crowding to become mature at the age of 7 to 8. So crowding is being fine-tuned later than acuity, but not as late as some of those estimates. And we can talk about some of the things that go into that, those differences later if you're interested. Okay, so having established this slow development, the question that occurs in my mind is whether the processes that we're seeing in these children in their fovea is the same process as what we see in our peripheral vision. Central to this kind of argument about crowding being an adaptive process is that it's providing the gist of things. And so this was something that we wanted to explore in this study that I'll talk about. So here we want to look at crowding in amblyopia. I mentioned earlier that this is the strabismic amblyopia is the disorder where you have this deviation of one of the eyes. This is a developmental disorder of vision that affects around 3% of the population, and it's defined by that ocular deviation. They have an impairment in acuity in that amblyopic eye relative to their fellow eye, their good eye. And you also see these strong elevations in the amblyopic fovea. So the question then is this the same process, this crowding effect that we see as what you see in those developmental cases that I just talked about, and in peripheral vision? And if that's the case, then that this shared mechanism predicts that you should see the same kinds of systematic errors in all of those cases, those predictable errors that I talked about that look towards averages and so on, should be present in all of these cases if this is a common mechanism. The alternative is that there may be random errors. There may be errors that are driven by metamorphopsia. So we know there are distortions of the visual field in amblyopia, which could produce a kind of shearing of the stimuli. And we also know that children, of course, have high lapse rates. They're inattentive at times, and so it could be that they're just coming in and out of the task and performing less well that way. And both of those things would produce random errors if they're periodically either distorting the images or coming in and out of doing the task properly. So what we did was very similar to what you saw before. We used the Vacman setup. Here we do it with stereoscopic shutters so that we can control which eye is seeing the target. We ran this with typically developing children and children with strabismic amblyopia, 20 of each, ages 3 to 8, so in that range where crowding is elevated. And we compare that to 20 adults viewing this, 10 adults viewing the stimuli peripherally. So the first thing we do is to measure acuity and crowding, as you saw in exactly the way we did before, where we measure acuity by scaling up and down Vacman in this setup, and then we make the ghosts achromatic and we scale the stimuli up and down. That tells us the size of the interference zone that we can work with, where we can put flankers within that to get these effects. And then we have a third phase here where there's an orientation matching task. So the story for the children is that the Vacman is being surrounded by imposters, and they have to find the real Vacman, who's always the one in the center. And the way that they do that is they tell us which way Vacman's looking by rotating a response dial, which controls a little element on a separate screen, and the amblyopic kids can see that with their good eye. And in order to tell us the orientation that the target is at, either in isolation or with flankers present. What we're going to do then is to fit the data with a population pooling model. I mentioned these models before, but this is our instantiation of that. If the element is uncrowded, you can represent that quite simply. So you just have a population of neurons that give you this Gaussian distribution, and there's a degree of noise associated with that. That's the first free parameter. The noise gives you some trial-to-trial variability, and so on this particular trial, we take the peak of that distribution, which gives us an 8-degree error. So this is all coded relative to the true orientation of the target. The targets were randomly oriented around the clock. When you then have flankers present, you have a second population response for those flanker elements with a separate noise input, noise parameter, so you can have more or less noise coming from the flankers. And then those two distributions are pooled in this stage when they're present with a weight that determines how strongly the flanker response dominates that final population. And in this case, with these very similar target flanker elements, this 30-degree case here, difference between the target orientation and the flankers, then what you tend to get are these assimilation errors in the model. So here there's an 18-degree shift that's intermediate between the true target orientation and the flanker orientation. But you can also, I didn't mention this before, but you can also get errors of substitution in these crowded arrays. So sometimes when you have very dissimilar elements, people can report either the target or the flanker, which has been called substitution errors in the literature. And the appealing aspect of these population pooling models is that they can account for both of these types of errors with the same process. So the reason that you get these substitution style errors according to these models is that now the target and the flanker distributions are very different. And so when you combine them, you get this bimodal distribution. And either because of the weights determining which of those is dominant, or because of noise giving you peaks on either side, you tend to get responses that are either around the target or around the flanker. And so in this case, there's a 90-degree error, which corresponds to the error, the orientation of the flankers, which is an error of substitution. So we're going to fit that to our data. This is the kind of thing that you see in the adult periphery. This is the average response for 10 observers. I've plotted this now relative to the true orientation of the target. So this is response error, and this is the frequency data. The gray circles give you the data from the 10 adults, and the black line gives you the best fitting population model. You can see for this uncrowded element that observers are very good at reproducing the orientation of that with a bit of scatter around that. And then when you add the flankers, you can see that there's this lower, broader distribution that's shifted to one side. So its peak, the peak of the responses here tends to be between the target value and the flankers, and the model does a very good job of capturing that shift. Whereas, as I mentioned, with the 90-degree flankers, people tend to report either the true target orientation here or the flanker orientation with this sort of bimodal distribution, which the pooling model is also able to produce. So you get these assimilation-like errors and these substitution errors in these different cases, both of which are well explained by that population pooling model. Here is the data from the 3 to 8 year old children with strabismic amblyopia. So they're looking at the stimuli through their amblyopic eye and telling us the orientation with their good eye. And here is the frequency. These are lower, noisier response distributions because these are kids after all. But you can see that they can do the task. They can reproduce that target orientation fairly faithfully when you ask them to, when it's presented in isolation. But when you add the flankers, you can see again, you get this lower, broader distribution with a peak that falls squarely between the target and the flanker orientations. Or when the flankers are very dissimilar, you get this bimodal distribution, this second peak in their error rates. So you get the same kind of errors in these cases of strabismic amblyopia, and the population pooling model again can capture the errors that are taking place there. Finally, here are the typically developing children. So these children have less crowding than the kids with strabismic amblyopia, but we're scaling the stimuli to fit them within their interference zones. And nonetheless, when you do that, you get the same kinds of errors. Again, you get they're able to do the task. So there's a sort of noisy distribution, but it's peaking at the true target orientation. When you add the flankers, you get this lower, broader distribution, again peaking in the middle, and then this evidence of bimodality here in the skew sort of secondary peak of the 90-degree condition. So the same kinds of errors, and again, the model can capture that pattern just by changing the weights that you apply to get different amounts of crowding in these circumstances. So the take-home message from this part is that these crowding errors are occurring not because we see nothing, but because we see the wrong object. That when you present an array of stimuli like this, that people see the target object as changed, either completely to match the flankers or partly, as in these kinds of assimilation errors. And this same process of pooling is evident in peripheral vision, in the typical visual system, in the amblyopic fovea, and in the developmental fovea, consistent with there being a common mechanism. So the final set of studies that I would like to talk about start to complicate this picture somewhat. So I mentioned in the beginning of this section on elevated foveal crowding that there's a rather diverse array of conditions where you see these elevations in crowding. And one of the more interesting ones in terms of comparison is the case of nystagmus. So here you have these excessive eye movements that are also present while you're making these tasks. And the question then is, do these eye movements alter the nature of the foveal crowding that we're seeing? Or can we understand all of this using a common mechanism here as well? So to give you a little more detail about infantile nystagmus syndrome, this, as I mentioned, is a syndrome that's characterized by involuntary eye movements. They tend to be primarily along the horizontal dimension rather than the vertical dimension. And so what you'll find when you ask somebody with INS to stare straight ahead is often they'll have this period of fixation followed by a drift and then a rapid jerk back into position. That's a jerk type nystagmus. Or at times these more pendular kind of movements where the eyes is more slowly moving back and forth, more smoothly moving back and forth over time. Related to the point about the heterogeneity of conditions that produce crowding effects, INS itself has quite heterogeneous origins. So you see a number of conditions that are associated with nystagmus. One of them is albinism, where you have a disorder of the skin pigment, of course, but where that also affects the photoreceptor pigments. Then you have, in addition to the eye movements, you also have retinal issues. So this is an OCT image of a typical retina. The cross section here shows you the foveal pit, which is where all of those photoreceptors are packed in and where you don't have any vasculature. So you get this kind of concentration of photoreceptors alone in this little pit here. But compare that with albinism, where you see this rather more flat distribution. So you don't get this foveal pit. It's what's known as foveal hyperplasia. And so that carries through to cortical visual regions as well, where you get a number of cortical deficits in terms of effects on retinopathy and so on. So here you have very clear retinal and cortical deficits that are present in addition to the eye movements. But there are also cases that occur in the absence of any clear pathology, and this is what's known as idiopathic INS or IINS. And so these cases tend to be diagnosed by exclusion, that once you've exhausted all of the other possibilities for what's causing the nystagmus, they're given a diagnosis of idiopathic nystagmus. And so here you can see here is an OCT image taken by my collaborator VJ, where you can see there's this foveal pit that's still present very clearly here, very similar to typical vision, so they're physiologically their visual system seems to be more or less intact, and yet they have this constant eye movement that's present. Now, it's clear that foveal crowding is elevated in these cases, but what prior studies have tended to do, because these kinds of patients can be hard to find, is that they've tended to chunk them together. So you see in a lot of these studies that people with albinism are grouped in with people with IINS. And so one of the things that we wanted to do was to separate them. We're sort of lucky here in London that we have Moorfields Eye Hospital, which is a big feeder clinic, and we get all kinds of patients coming from all around the country. And so we're able to get samples that are specifically from these idiopathic groups and these albinotic groups, which I'll show you some data from. The question then is what drives crowding in these cases? So could these elevations in crowding reflect some kind of cortical deficit like we saw in amblyopia, or the cortical changes that we saw in development? Or is there something about the eye movements that could change things here? Could there be something to do with the image motion or the smear that produces these effects? You could imagine taking an array of a target surrounded by flankers and then moving it across the retina rapidly, that you tend to get this kind of smearing over time, kind of like the temporal crowding effects that Jaffa has worked so extensively on. There you could get these kinds of effects over time rather than these instantaneous effects that we've been talking about, or you could get just a kind of masking effect where the flankers simply overwrite the target. If that's the case, if there is an effect of image smear here, then you really should see greater disruption on the horizontal dimension, because the nystagmus waveforms, as I mentioned, are predominantly horizontal. So if you were to take a target and surrounded by flankers, then when you move the eyes back and forth, left and right across that array, the elements are smearing into each other. Whereas if you have vertical flankers, then they're all moving in tandem. And so you should get greater effects of crowding on the horizontal dimension than the vertical if this were something that's primarily driven by the eye movements. So what we did was simply that, to measure the shape of those foveal interference zones in typical adults and to compare that in cases of amblyopia and idiopathic nystagmus. The procedure that we follow is more or less exactly the same, more or less the same as what you saw earlier with the Vacman elements. Here, they're adults, so we are using Land.C stimuli, but we're doing the same kind of scaling approach. They tell us for AFC which way the gap in the C is facing, and we scale those elements up and down. Then you can see this is example data from a single observer, the unflanked performance here in olive. Then these are the conditions with horizontal and vertical flankers. You can see this individual is very strongly affected by the presence of those horizontal flankers, so they need a larger gap size as a threshold in order to perform that task. And so we measured these thresholds in typical adults, in adults with strabismic amblyopia, and in 8 individuals with idiopathic nystagmus. These are the individuals with the unexplained nystagmus without clear cortical deficits. And this is what we find on average. So in the control participants, this is foveal crowding. They're looking directly at it. Foveal performance. And so the unflanked performance is very good. It's below 1 minute of arc for the gap size. So they're very clearly able to see these elements. You add flankers and you get a little crowding effect. That's consistent with a lot of prior studies, but it doesn't really matter whether the flankers are present on the horizontal or the vertical dimension there. Compare that to amblyopia here. You see this very clear elevation in thresholds. So for the unflanked elements, that's their acuity deficit. And then you add flankers and they become substantially worse now. Although there's a bit of a drop here when you add the vertical flankers, that's primarily driven by one observer. The other 9 observers showed no difference, no reliable difference between the horizontal vertical dimension, and indeed the difference between these conditions is not significant. But compare that with the individuals with idiopathic nystagmus. Here you can see that there is an elevation in performance with the unflanked element, and then you add the flankers, the horizontal flankers, and they get worse. But performance is much better when you have just the vertical flankers. So there is a clear horizontal elongation of that interference zone, which is suggestive of an effect that's very strongly driven by eye movements. So if that's the case, if this elevation in nystagmus is driven by eye movements, then we should be able to produce equivalent effects just by simulating these effects in typical observers. And so that's what we did here, is to take these stimuli and to move them around the screen while observers fixated or moved around the stimuli. We did both. That here we're using the waveforms taken from our nystagmus participants and presenting the stimuli to typical observers. If this is something that's purely driven by the motion of the stimuli, then you should be able to get the same pattern. So now we have 10 observers viewing the stimuli with and without these waveforms. And this is what we find. The typical observers with no motion, again, have very good performance for the unflanked. They get crowding effects when you add the flankers, but there's no difference between the horizontal and the vertical dimensions. But when you apply this stimulus motion, you get a very small elevation for the unflanked, but a very clear elevation with the flankers, and a difference on the horizontal and vertical dimensions. And these dashed lines here show you the level of performance that we saw in those individuals with nystagmus. And you can see that we can get pretty close to their deficit on the horizontal dimension and the vertical. Interestingly, we're not able to reproduce their performance in the unflanked case, so we can't completely exclude the possibility that there's some kind of cortical deficit here, that there are changes in the brain related to the condition here. But at least in the case of crowding, there's a very clear contribution from eye movements, that it's very hard to it would be very hard to exclude the idea that this is purely driven by eye movements. We can compare that in our recent study. We've been comparing that with individuals with albinism. So recall that these individuals have both foveal hyperplasia, so those retinal deficits, and cortical deficits. We can ask then, what does the shape of these interference zones look like in that case? Can we find here clearer evidence for a cortical deficit that's driving crowding, or is that also just purely driven by the eye movements? And so here we do the same thing. We measure the shape of crowding in adults with albinism and compare that to those age-matched controls. This is the data that we get, exactly the same procedure. Again, the controls show a little bit of crowding and no difference on the horizontal vertical dimensions. This is the data that I showed you before from the idiopathic nystagmus cases. And here are the adults with albinism. So you can see their deficit for single elements is much greater. That's driven by the foveal hyperplasia and things like that. But you add the flankers and they get worse, so they have a very strong crowding effect in the fovea, and then but you switch that to the vertical flankers and the deficit is much reduced. So the horizontal elongation of the interference zone here remains clear, but the overall level of performance here very clearly shows that there is an effect of a cortical deficit here. So that if you recall, the simulated nystagmus was down here as well with the IINS data. And so eye movements alone can't produce this level of deficit, suggesting that there is something more there to the level of crowding there. So when you compare across these conditions, the extent of foveal crowding, you see that in typical adults and in strabismic amblyopia, if this is the smallest kind of letter that you could recognize, then you have this sort of interference zone around that's fairly small. You could just about fit flankers within that will disrupt that. But when you go to strabismic amblyopia, you need to make the target itself bigger, but also much more separated from its surrounds, that you get this large interference zone in strabismic amblyopia. In the case of both idiopathic nystagmus and in albinism, you get this elongation, that you have this horizontal stretching of the interference zone, which is demonstrative of an effect from eye movements, which we can also replicate with that simulated group, so show that these effects are applicable there too. Overall, though, the levels of crowding that you see, both in terms of the level of deficit that you see for acuity in particular in idiopathic nystagmus and the overall levels of crowding in albinism, do suggest that there's likely to be some additional cortical deficit. And so the message to take from this is that these crowding effects can derive from multiple sources, most likely. So, in the interest of time, I'll summarize there. So, crowding, I've argued, is the primary limit on our peripheral vision, with foveal elevations in development in amblyopia and in nystagmus. Those foveal elevations produce the same kinds of errors in peripheral vision as you see in typical adults, suggesting a common process of pooling. But then there's also these elongated interference effects in nystagmus, which suggests that multiple factors can produce these effects. That when you look at these diverse conditions, you have to consider multiple sources for these kinds of disruptions. Nonetheless, what I think all of this shows is that crowding is a process that works to simplify visual input. It gives us the gist of what we see, either when neural resources are low or when the inputs are degraded in some way, so over time with those eye movements, for instance. And what it suggests to me is that crowding is something we can think of as the default state of the visual system, that it's something that's present in the absence of any fine-tuning, that it develops slowly relative to acuity, and that maturation leaves it particularly susceptible to disruption, but that this is a core process that's driving vision in the periphery, and that it also needs to be honed extensively in our fovea. So, thanks for listening. Thanks for your attention. Thanks to our funders. These are my collaborators who've worked on these various projects: Annegret, Tessa, Alexandra, Vijay, Maria, and Marilia. And happy to take questions. Thank you.
Thank you so much, John, for a very, very interesting talk. And I want to invite everybody to unmute yourself and give John a really big applaud. Really, really interesting. If
Thanks.
you are happy to take questions, which I
Of course,
understand you
yes.
are. So I'll start. I understand that probably there are additional questions, but I want to ask about the last results that you showed about the nystagmus and then also about the albinism. I mean, you showed
Hmm.
that the if I understand correctly, that the horizontal motion is also now in the controls. Uh, in a way, I'm thinking about this that there is no efferent copy because they are not producing the motion. So, and usually when we move the eyes, we have, hypothetically, let's say, we've got the efferent copy. So I'm wondering if that implies that maybe nystagmus and albinism also don't have an efferent copy for these motions. Just
Yeah, there is
thoughts.
something to do with difference copy bundled in there. So one of the symptoms in so there's you can also get acquired nystagmus, right? So if you have an accident or damage to the superior colliculus and places like that, you can get acquired forms of nystagmus, and people with that form of nystagmus experience oscillopsia. So they experience the world as shaking and wobbling with their eyes.
Hmm.
And but people with infantile nystagmus who've had this from birth don't. They
Oh, they
are
don't.
they see the world as stable even though their eyes are
Hmm.
constantly
I
moving like
see.
that. So there is something in there related to the efference copy for sure. And I think it's that kind of adjustment. I mean, it could be that kind of Bosco Chan has a study where or a model where they argued that in typical vision, crowding was related to the kind of saccade statistics that you have, and I'm not completely convinced by that in typical vision, but I think in these cases where you have these eye movements, that it could be this kind of adjustment over time that you're pooling over this larger region in order to take in that information and discount the eye movements, perhaps. Yeah, I think that could be part of it.
Okay, great. Really, really interesting. I'll continue. But if there are additional questions, I'll put myself aside and let others ask. I wanted to ask about the horizontal kind of like receptive field that you showed. So basically, if you have it elongated in the horizontal, that really has a, substantial influence on reading
Mm.
because you need this integration window, and if there's an interference that is bigger on the horizontal, then you are likely to have a problem building up, templates that are built up of multiple elements, or maybe you have more confusions. So is that, I mean, I know these populations do have really issues with learning to read, and they need to magnify the text, and I don't know how to what kind of levels they get with respect to, but I assume this is a very big challenge.
Yeah.
Um,
Yeah, absolutely. And it's trickier than you might think to resolve. So there's where I see the most work on this is in the context of dyslexia, where there was a study 10 or so years ago showing that if you have dyslexic children and you scale the stimuli, so you space the stimuli more widely, that you increase the spacing between the letters, that the reading speed would increase. And so then the idea was that you have to overcome these crowding effects. But then my understanding is that it's been difficult to replicate that. There's a set of failures to replicate that.
Hmm.
And the argument there is that what you're doing is when you space the letters apart, even if you do it proportionally to the space between words, you're still messing up the low spatial frequencies and things like that that are so important for moving your eyes through the block of text and things like that. So there's not great evidence that you can overcome these effects easily within a block of text. But yeah, it absolutely there are correlations between in amblyopia, for instance, the level of deficit and reading speed are correlated. Dennis Pelly's shown that. So yeah, it absolutely has an effect there. Yeah.
Okay, thank you. Great. Are there additional questions?
Let me ask one.
Yeah, sure.
Is the foveal crowding in strabismus proportional or correlated with the squint, the size of the squint, the side of the squint?
Good question. Yeah, it is affected by the direction. So esotropia, where the eye goes inwards, is worse than exotropia, where the eye goes
Mm.
outwards. And I think the idea there is that the it's about where the fovea gets the interfering information from in terms of the binocular overlap. Um, like you're moving in towards the other eye as opposed to away from the other eye. So that gives you more crowding than in esotropia. We've tried to look at the level of deviation previously, and it's at least not a linear function. So you do get sort of micro squint. What do you call it? Very small angle deviations, which will typically be less affected than people with very large deviations. Um, but beyond a certain level, it doesn't seem to matter what the deviation is. It's just bad. So it's not a linear function at least. But yeah, I think it does matter
So
somewhere.
does this tell you what the reason for this crowding, what the mechanism by which the crowding developed?
Yeah, I mean, I think it's indirect in the sense that the binocular conflict that you get when the eyes are misaligned changes the distribution of neural sensitivity. So it means that you get fewer binocular neurons because they're not able to make those connections, um, and then you get this diversion of resources away from the amblyopic eye because it's blurred and things like that, and that it's that drop in neural population numbers that drives the crowding effect. I would argue that's my hypothesis at least, and so the crowding is a response to the neural deficit rather than the cause in that sense.
Excellent. Yes, thank you.
Okay. So, John, I really want to thank you a lot for joining us and for presenting such interesting and wonderful research. Really, my hands are tied because my okay, well, anyway.
You're holding your laptop, yeah.
Yeah, I
Thank you.
guess that's why it's moving so much. Yeah, okay. So I really want to thank you so much for joining us and presenting such interesting and fascinating research. And we will be taking, I'm just now saying, we will be taking 2 weeks off because I mean, it's Passover and I assume it's also Easter vacation. Is that
Yeah.
true,
Yeah, soon.
Kieran?
Yeah.
Okay, great. And we'll be back after that with a lot of exciting talks afterwards. So have a lovely rest of the week and vacation, and may peace guide us through the planet. Yeah.
Stay safe. Yeah.
Yes. Thank you so much.
All right. Lovely
Thank
to see
you.
you all. Thank you for
Lovely
inviting
to
me
see
again.
you too.
Yeah.
Bye bye.
Cheers. Bye.
Cheers.